All right, everyone, we are right on time. So I want to make sure we can kick this off. We have a lot of really exciting things to talk about today. You're basically getting a 3-in-1 presentation, which is kind of cool. So welcome to day 3 of Invasive Species Awareness Week. Hopefully you are here for the Aquatic Invasive Species talks. And we're really excited to be able to host this today. I'm going to go through, just to kind of give you the run of show, I'm going to go through a few housekeeping items. I'm going to explain how the pesticide credits are going to be working today, and then we'll get right into the meat of the topic because I know that's what you're all here for. So just to get things started, my name is Alexis Hafley. I'm the Education Outreach Coordinator for the Washington Invasive Species Council, and we are co-hosting today's webinar with these fine folks from the Department of Fish and Wildlife. We're really excited about this one. It's going to have a lot of excellent information in it. This is a Zoom webinar. So a Zoom webinar is a little bit different than a Zoom meeting, which I feel like probably most of us are familiar with by now. And some of the differences come as an attendee. So if you've noticed, if you've taken a look at your taskbar at the bottom, you don't have the ability to unmute yourself or have your camera shown. That's totally normal. That's part of the webinar series. But what you can do is you can write in the chat box, or you can use the question and answer feature. And that's how we're going to be interfacing with you as the presentations go on throughout the day. This webinar is being recorded. We will have it posted to the council's YouTube page, probably early next week. So keep an eye out for that. We're going to be sharing some of the links on social media, as well as sending out an email blast. So if you have anybody that would really love this information, or you found this webinar really valuable and you want to come back and reference some things that you learned later, that will be available for you. If you're having any issues and you need any help, or you can't hear us or see us, or anything's kind of wonky, just leave a message in the chat and somebody will be able to help you kind of troubleshoot any issues they have as soon as they can. And then should the webinar fail, fingers crossed, but you never know, should the webinar fail, everybody will just give it a couple of minutes. Hopefully everything will reset, and then we'll all try logging back in to pick up where we left off. And then lastly, we are hoping to have a few minutes at the end for some question and answer period. So as the presenters are going through, if you do want to leave a question, I would encourage you to do that as they're talking about it. And hopefully we'll have a few minutes at the very end of the presentation to get to those. And then for you, those of you wanting pesticide recertification credits, if this isn't you, feel free to let your attention wander. But if you're here for the pesticide recertification credits, this webinar is worth 1 recertification credit. And the way that you're going to get that credit is towards sometime during the presentation today, we're going to have a code word slide. So if you're just now logging in, don't worry, you haven't missed anything yet. It will be really obvious when that code word slide is. We're going to have an entire PowerPoint slide on it. I will announce it. I'll repeat what the code word is. We'll have it up for a couple of minutes to give everybody a chance to type it in. When you see that code word and you're prompted, please type the code word into the chat. That's how I'm going to verify your attendance for today's webinar. And then after the webinar is over, everyone who registered is going to get an email with further instructions. So in that email, that's where we're going to get some of that information that we need from you to confirm. So your name, your pesticide license number, all of that's going to come after the webinar. So make sure to keep a lookout for that. And without any further ado, welcome to the Aquatic Invasive Species webinar. And I'm going to pass it over to Amanda, who is going to get us kicked off. So take it away, Dr. Amanda.
All right. Just going to share my screen here. Can everybody see that okay?
Yes, it looks great.
All right. All right, good morning. So my name is Amanda. I'm an environmental specialist with the Aquatic Invasive Species Unit, and I'm just here to start out this session by giving a brief overview for everybody of what aquatic invasive species are, how they get here, and what the Aquatic Invasive Species Unit is, and in very general terms, what we do. So aquatic invasive species are species that do not naturally occur in a particular region and that cause measurable harm to native species and habitats, or to the local economy or to public health. And aquatic invasive species are invasive species that inhabit fresh and marine waters. Going into some more detail, invasive species, including aquatic invasive species, or AIS, move primarily via human-mediated pathways. So human activities are what brings them here to Washington or to whatever part of the world they're invasive in. Some of the most common ways that aquatic invasive species arrive to Washington include the growth of aquatic organisms on equipment and vessels, and this is called biofouling. And that's what these workers are here cleaning the whole ship. They're cleaning off biofouling that could contain potentially invasive organisms. They may also hitchhike with desirable farmed aquatic species. So these are giant Pacific oysters, which are native to Japan. And when they were first brought to the west coast of the United States for farming, they came with some hitchhiker species, sponges, tunicates, that then became invasive here in the United States. Ballast water is something else that large commercial vessels may carry, and it's water that helps them maintain their buoyancy and stability in the water while they're underway. But when they let that ballast water off, that might have organisms entrained with it that you can see in this picture. This is a bulk carrier that is letting off some ballast water. And that ballast may have propagules or whole invasive species entrained in it that it may then release. People do sometimes release organisms unintentionally when they clean their aquariums, or they may even release them intentionally, believing that, you know, either they don't want the animal anymore, or they believe that the animal will be happier in the wild. And species like this rosy red minnow have become invasive in Canada as aquarium escapees. It's not clear whether it was an accidental or intentional release. And to be classified as invasive, species have to cause measurable harm. And this harm is generally classified as ecological, which is harm to native species and habitats; economic harm, which is harm to commerce and infrastructure; and public health, harm to public health. So that would be pathogens or parasites that get out, establish a population, and then can become dangerous for human beings. There have actually been some cases of cholera linked to ballast water discharges in the southeastern United States. And this is an example, this picture is an example of harm to a native species. This is a picture from the Midwest, and all of these little triangles are little dracinid mussels that are invasive that we'll hear more about in detail. So zebra and quagga mussels are two of the species of concern that we're going to be hearing a lot about today. And in this case, they're overgrowing another bivalve or another organism. And when that happens to organisms with like mussels and clams, they can't open up their valves, they can't open their shells, and so they can't breathe. This animal looks like it may already have smothered. So here are a few examples of AIS that have been getting some press lately in Washington State. We have fish like this northern pike, and amphibians like this African clawed frog. And we also have invertebrates as well. European green crabs are a species that we're going to hear a lot about today. They've been found on the outer coast as well as in northern Puget Sound. Zebra mussels, again, this is the same picture seen on this other large freshwater bivalve, are a species of extreme concern in Washington. We're going to be hearing about monitoring efforts to watch and see if they're arriving in Washington, because they are easily transmitted on recreational vehicles, and they've decimated ecosystems and clogged water pipelines where they are invasive. In addition to these large, somewhat more obvious species, it's important to remember that AIS include planktonic life and microscopic life as well. So tiny invasive plankton often outcompete native planktonic species, and this can starve fish that rely on the native species for food. And then there are meroplankton, which are the tiny planktonic larvae of animals like worms and barnacles and clams and crabs. They can also be introduced to bodies of water where they will afterwards metamorphose into their adult forms and go on to establish invasive populations. So I think the thing that I want to really focus on in my talk is that invasive species are typically managed in two complementary manners, and both of these approaches are essential for protecting native ecosystems. Rapid response or emergency response, these measures can be mobilized to eradicate or knock back an AIS that is detected in the field. This requires multi-agency coordination, so a lot of work at the front. Rapid responses have to be effective in reducing or eradicating the localized populations of an invasive species, but they also have to be safe for humans and other species in the affected area. So this is a lot of coordination that has to happen beforehand. We have to know very clearly what we're going to do when we find that first individual or those first few individuals. And often rapid response has to be followed up with monitoring to determine whether measures were effective at actually knocking back that population. So given all of these elements, rapid response is typically very expensive, but it also comes with a very high and obvious impact. So you've got an AIS outbreak and it's controlled in a localized area. Prevention, on the other hand, requires that we find ways to control the pathways by which AIS arrive in Washington. So this requires continuous monitoring of these pathways and review of the best available science. Also, overall prevention tends to be less expensive than rapid response tends to be. And it's often difficult, however, to get political and monetary support for these kinds of efforts because the impacts are a lot harder to see. We're asking for a lot of support up front before an organism becomes a problem. So let's look at how the AIS unit approaches both prevention and rapid response. So I want to start with prevention. And so again, that is controlling the pathways by which organisms might arrive here. We conduct direct inspection of commercial vessels and recreational vessels to ensure that these vessels are properly managing their hulls, their ballast water if they have it, those kinds of vectors that in order to remove any potential aquatic invasive species. So these first two pictures, an inspector working with a bridge crew on a large marine-going vessel to go over their ballast water management plan. So how they're managing that ballast water to make sure they're not releasing any of those potentially entrained invasive species. There's also monitoring that's going on, taking samples of vessels. So that's what this inspector is doing here, who's holding that plankton net. He's actually taking a ballast water sample. And then there are also inspection and decontamination stations around the state for recreational boats to both check that they have properly managed their vessel and are not carrying, say, any zebra or quagga mussels, and that if anything's found, they can be decontaminated before they go into the water here in Washington. And this is a really cool picture of, actually, we're using some very novel approaches. One of them is Puddles, the dracinid sniffing dog, so trained to sniff for zebra and quagga mussels on boats. And the best thing for all concerned parties is that everyone be educated, right? Everyone be educated in the threats surrounding invasive species and how to manage their vessels to prevent the release of invasive species. And education and outreach efforts include signage and other outreach materials like these that encourage recreational boat owners to clean, drain, and dry their boats before moving them between bodies of water. This is a method that's very effective at preventing the movement of AIS into and around in Washington state. It's one that's most important at preventing zebra mussels from entering our state. We're going to hear, like I said, we're going to hear a lot more about zebra mussels monitoring and outreach efforts for that later today. So there is still the chance that invasive species are going to make it through all of these prevention measures. So a great deal of the AIS unit is dedicated to coordinating rapid response plans and executing those plans when needed. So time is of the essence. This is the thing to remember with rapid response. Time is of the essence when invasive species are detected in our state. we have to assess and respond properly to the specific AIS, the specific invasive species that was detected. But any time spent evaluating the problem and deciding what to do is time that that population is growing and its impacts are growing. So before the fact, we coordinate with agencies that have the scientific and logistic capacity to respond in an emergency. Recently, for example, green crabs were found near the northern boundary of Puget Sound. Our field experts knew that it was only a matter of time before rapid response measures would likely be needed here in Washington state, specific to European green crabs. So something that was actually going to work for the specific biology of the species. And the AIS unit worked with Sea Grant Washington and the Puget Sound Partnership, and also agency partners at Canada Fisheries and Oceans. This was in 2019 to develop a rapid response plan. And that was specifically tailored to the event of a green crab invasion in Puget Sound. And sure enough, some of the first European green crabs were detected by citizens in the San Juan Islands. And the established plan was put into action and made it possible to start removing some of those localized populations. And also to set up, most importantly, a monitoring program to determine whether these measures were effective and where they might next be needed. So to wrap up and take us into really the meat, I've talked a lot about European green crabs and zebra and quagga mussels. This is we're going to be hearing a lot of the details about some of those species of extreme concern here in Washington state. And so to wrap up and take us into those, really, I just want, what I want everybody to take away as 4 points from my introductory talk is that, first of all, aquatic invasive species are a threat to Washington state. They are species that do not occur here naturally, and they cause measurable harm to species, habitats, and people here in Washington. Aquatic invasive species arrive via these pathways that are human mediated. So it's human activities that are moving them around, human activities that we have to be watching and being very careful of. And our AIS unit at the Washington Department of Fish and Wildlife is using both preventative and rapid response measures to address AIS in Washington state, to monitor and look for, where are we going to find some of these populations? Where are they going to first pop up? And what do we do when we see them? But most importantly, how do we stop that from happening if at all we can? Most importantly, agencies and concerned citizens can find us here at this link. And there's a lot of information there about invasive species in Washington, what to look for, and some of the activities that are going on right now in our unit. So hopefully I think these slides will be shared at the end of today, or maybe we can make this link available in chat if people just want to have a little more information about other species and things that they might not hear about today. So thank you very much. That is my talk, and I will be around at the panel later. And if there's time for questions now, I can also answer some now. So I'm just going to stop sharing my screen.
Perfect. Thanks, Amanda. And watch us move around. We're doing things and sharing screens like we've been doing this forever.
And thank you, Alexis. Found more cool pictures, and so Alexis allowed me to share my screen so I could show my cool new pictures.
Yeah, my pleasure. All right, Eric, you are up. Take it away. And whenever you want me to advance, just say so.
Okay, thank you. I'm Eric Anderson, and I obviously am with the Washington Department of Fish and Wildlife, and I'm a captain with our enforcement program. And I operate and manage our watercraft inspection program to try and prevent, again, prevention is the key on this, from trying to allow aquatic invasive species coming into our state. And more specifically, my focus and my program's focus is trying to keep zebra and quagga mussels out of Washington and the Pacific Northwest, because these are, I believe, the single biggest threat to not just the environment in the Pacific Northwest, but also to the economy and to the citizens of us. And just like this slide just says, it is the biggest AIS threat to the natural resources. And it's funny because being with Fish and Wildlife, you'd think I would focus mostly on you know, the natural resources, but I also want to focus in big bold letters there, the economy, because these things would have an impact on every single citizen of the Pacific Northwest should they get in here. So go ahead and next slide. So what is a zebra and quagga mussel? A zebra and quagga mussels, they are little, as Dr. Newsom talked about, they're called dreissiniid mussels. They are a small bivalve freshwater mollusk. They didn't originate here in the Pacific Northwest. They actually were over in Europe in the Black and Caspian Sea, which is mostly a freshwater system, even though they're called a sea. We're not too, we like to describe things as they look. And so they were originally dubbed zebra mussels because they have those alternating stripes on them. So they actually have zebra stripes on them. They can get a little dark, but the stripes are very pronounced on them. And these things are not very big. A old adult that's lived for a few years might get up to 3/4 of an inch in length. But juveniles, the small ones, are about the size of a sesame seed. Also, when we're inspecting watercraft, the veligers, or the little larva form of them, if they settle on the outside of a hard surface, if you brush your hand against them, they actually feel like sandpaper on the surface. So one of the things we're going to talk about is the inspection program, and we actually inspect both visually and tactily. We feel the hull of the boats to make sure that we don't have the larva or the veligers settling on them. So next slide. So what makes these things unstoppable invaders? And I've coined the phrase that these things are the cockroaches of the shellfish world, i.e. these things are super adaptable. They're able to live in such a wide variety of aquatic environments that it's absolutely amazing. They will not live in salt water, but they can live in brackish water. So they can actually tolerate a various level of salinity. They can also tolerate a wide range of temperatures. You'd think that when you're starting to get freshwater temperatures that are up into the 70-degree and higher, that most aquatic organisms perish in that, they die. Zebra mussels do quite fine. Zebra mussels also can live in water that gets almost down frozen solid. So they have a super high survivability rate. Wide range of areas that they can get. The other thing that is absolutely astonishing to them is that they have an astronomical reproductive rate. So when they spawn a pair, male and female pair, they can spawn up to 1 million veligers in a spawning cycle. And generally, when you have something that has that high of a cycle, you usually have a high die-off. Not with these things. These things actually have a really high survival rate. Almost a third of them survive. So up to 33% of 1 million of these individual mussels can survive. And so for some of us old folks might remember there used to be a commercial talking about, you know, you tell 2 friends and they tell 2 friends and so on, and that exponential growth. Well, imagine if you had 333,000 friends and they told 333,000 friends, and you can imagine how fast these things take over. The other thing, when I was talking about water temperatures on it, water temperatures actually also control the number of spawning cycles that zebra or quagga mussels can go in a year. And so when they hit an optimal range of, it's between about 45 to 55 degrees, that's really their sweet spot. And if they hit temperatures like that, they don't just spawn once a year. They can actually have numerous cycles. And there's actually been some places in the US where they've overtaken, and when they've been doing the monitoring, they actually don't find a period of the year where there's not veligers in the water. So they're continuously spawning. So this makes them, like I said, an unstoppable invader. And I'm going to give you a picture on this. Didn't have time to put up a slide, but I want everybody to remember back to high school geography and U.S. geography, and picture the continental U.S. and look where you have the Great Lakes and the Mississippi River region. And so this will tell you how fast these things can take over. We discovered them in 1988 in Lake St. Clair, which is a little connector lake between Lake Huron and Lake Erie. And so they figured they came over in through the ballast water on some of the commercial traffic going into the Great Lakes. So we discovered them. And we're like, what are these things? They keyed them out, figured out they were zebra mussels. But they'd obviously been there for a short period of time before we found them. And this was 1988. By 1993, they had completely colonized all the entire Great Lakes. I mean, and when I say colonized, I mean these things had taken over. And they had spread throughout the Mississippi River region because it is connected to the Great Lakes. So think about that. In 5 years, they basically took over the waterway of the Eastern America. 5 years. That is an exponential takeover. From 2004 to 2006, because we were trying to stop them, we actually slowed their westward progression a bit. But then in 2007, we had our lightning strike in the Southwest, and they were discovered down in Lake Mead, which is in the Colorado River Basin. So basically a 1,000-mile jump, and now they're knocking on the door of the Pacific Northwest. And so that's why we're highly looking for these things, trying to keep them out. Next slide. So the Columbia River, which is the cornerstone of the Pacific Northwest, the roll on the mighty Columbia, it has a huge target on it. It is the last great river basin in the US that does not have these things. And so next slide, we're going to talk about what we're starting to be at stake with these things. So imagine, because these things reproduce and attach to things, so they colonize in these dense mats and clusters, they actually start completely filling in surfaces of pipes, filters, screens, you name it. And these are some of these pictures show you how they've infiltrated and got into mechanical systems, and they start just completely blocking and diminishing the flow rates of these mechanical systems. Next slide. And so here's the big ticket item on the Columbia River, and it is our federal hydro projects. We have over 29 federal hydro projects throughout the Columbia River Basin, and that includes not only like the U.S. Army Corps of Engineers, the hydroelectric for Bonneville Power, all of the hydroelectric projects, but also things such as the Bureau of Reclamation, water projects, so irrigation systems. Next slide. So in the hydroelectric dam, this is, so you saw those pictures before. What is in a hydroelectric dam? You literally have miles of pipe. You have screens, filters, pumps. These things all have, obviously the mussels attach on these hard surfaces, and they start completely shutting down the hydroelectric facilities. And so they actually did a study in the early 2000s and calculated out that if zebra or quagga mussels got into the Columbia River system, it would, back then, it was about $25 million a year per hydroelectric project to control these things. So if you can imagine 29 projects and $25 million each a year, that's a big ticket, and it would completely trickle down to the citizens of the Pacific Northwest, and you're going to have huge electrical bill costs for these things. So hydroelectric dams, big thing. Next slide. Other thing along the Columbia River, again, I'm saying this will affect all citizens. Municipalities, we pull out of the Columbia River system drinking water. And so here's some pictures of some of these drinking water facilities. And again, what do we have? We have miles of pipe, we have filters, we have screens, we have everything that these things would clog up and literally come to a standstill. There are ways to manage them in there. As I like to call it, they can crank up the chlorine on your drinking water. That makes your drinking water especially delicious. If anybody has ever got a mouthful of pool water at a YMCA, there's what your drinking water is going to taste like if we have to try and control these things through chlorine feed systems. So back in the Midwest, already they're dealing with them, and the water companies back there literally have to pass it on to the people that buy from them. And it's millions and millions of dollars a year to try and control and deal with these things. So again, high water costs. So again, the economy, this is trickling down to the citizens. Next slide. Now let's talk about the backbone of the Pacific Northwest, which is our agricultural community. In the state of Washington, 97 irrigation districts alone that pull water from the Columbia River. And that's serving hundreds of farms. And again, this would affect from the intake pipe pulling out of the river with the screens, the pumps, all the way out to the spigot out in the fields. You can imagine the devastation this would cause to the farmers. Again, they would be having to replace their pumps and do things on a yearly basis with maintenance. So again, agricultural costs would go way up. Big damage to our industry and farming. Next slide. And now I'll get into, since with fish and wildlife, the biological and recreational impacts. Again, one of the things, all these dams that we have out there, they have fish bypass facilities to protect the endangered salmon of the Columbia River. And if these fish ladders and these things fill up with these mussels, they have sharp edges on them. So all of a sudden, these fish bypass facilities, which are supposed to be protecting the salmon, trying to get them past, going upstream to spawn and back down the smolts heading out, basically, we're going to turn those things into Cuisinarts for the salmon if these mussels get there. So the adults are going to get beat up as they're trying to go through the fish ladders, getting damaged. It's going to have a mortality on them. And then the smolts going back out the other direction. Again, we already have a high mortality rate on. They really can't take another shot on this, but it would be bad. The other thing that these things do to other critters in the water is they actually filter so much water, they disrupt the natural nutrient cycle in the water bodies that they inhabit. They take the zooplankton out, they take the plankton out, for phytoplankton out, and so they disrupt all the other. They completely turn the ecosystem that they invade into their ecosystem, and it's really hard on the native animal and plants there. Next slide. So how are they going to get there? I love Gary Larson. This isn't far from the truth of how they would get here. They're going to get here on recreational watercraft, and that'd be the next slide. This is how they have made it across America. They actually attach on the outside of towed recreational watercraft and commercial watercraft that goes through our highways. These things attach on the outside of boats, and if people don't know that they're on there, and as we have our moniker, clean, drain, dried them, they can transport them across the states. And these things can actually live out of water for up to 30 days. So, I mean, that's a long time. They close up, they stay moist. If they're in a cool, moist environment, they survive. Somebody comes up here, backs into the Columbia River, goes for a day of water skiing, and we're in trouble. Next slide. So what we're trying to do to stop this is we currently have 2 watercraft inspection stations. I know that doesn't sound like a lot, but we're building the program. They are both at the entrances in Eastern Washington, one on I-90 at Spokane, coming in from Idaho, and the other one is just outside of Pasco on Highway 395. For 2021, we were able to open and get enough funding that both those stations are going to be open year-round this year. We are up and actively checking boats right now. It's funny, we still have boats traveling around in the dead of winter. They're coming in from the state, from other states. Next slide. This will give you an idea of how we're trying in the growth of this program. In 2017, between the 2 check stations, we didn't have a lot of funding. We were only able to keep those open 216 days between the 2 of them. And that's really kind of hard to see, but we inspected 9,054 boats that year. And as of, and it should say, I see a mistake on the slide. It should say as of 2020, between the 2 check stations, we had 645 check station days, and we inspected physically, hands-on, our inspectors inspected 32,735 boats last year. And out of those 32,000, we had 25 boats that had mussels on them that we dealt with and had to decontaminate and make them so they weren't a threat to our waters. So biggest year ever. 2021, And during the COVID, we have not seen a slowdown. We've actually seen boats. People can't go to the movies, can't go to concerts, but they can go out and put their boats in the water. So there's actually a little increase on that. Next slide. Luckily this year as well, we have a nice grant from Bureau of Reclamations, and with their generous grant, we are opening up our third check station. It's on I-90 as well. It's checking boats heading eastbound into the Columbia River Basin. We're catching boats that will come both from north out of Canada, down I-5 and turn east, or coming up from the south out of Oregon, heading up I-5 and then turning on I-5 or on I-90 and heading into eastern Washington. So this is providing, we got protection on the east end, and now we got some on the west end. And hopefully this is a pilot project for 2 years. We'll be able to find funding to continue it on and expand the program. Next slide. And then finally, the other key on this, as we've talked, is promoting the education and the outreach with the public. It's not rocket science to prevent these things from, going to new places. Boat owners, we want folks to clean, drain, and dry your boats. If they pull out of the water, they wipe off the hull, drain the water out of the holds on it, and remove all the vegetation before they head down the road. They've just stopped the problem in its tracks. So that's our huge outreach campaign. It's a real easy one to remember. And then, like we were talking about, the other picture there, that is, as I call it, Puddles the Wonder Dog. Puddles is our mussel sniffing dog, and she's absolutely amazing. She's handled by Sergeant Pam Taylor. And she goes to both our check stations at various times, and she is trained to sniff mussels out on the boats. And the benefit to having a dog, if we have our human inspectors do a boat, it takes anywhere from 3 to 5 minutes to visually and physically inspect a boat as a human. Puddles can inspect a boat in about 30 seconds. Her nose is amazingly strong, and she's a whiz when it comes to it. So we also have the hashtag "It's Not Rough" to stop these things. And so with that, I think that's my last slide.
Yeah, thanks, Eric. All right, we're going to jump right in. Richard, you are on deck. And then same thing, if you want to tell me just when you want me to advance the slide.
Okay, will do. Sounds good. My name is Richard Visser. I've been working with Aquatic Invasive Species for a while now, 2016, I believe. And I'm here to talk about management of invasive species and how we kind of deal with that. Next slide. So we kind of talked about this already, but some of the big species that I'm going to be covering is zebra and quagga mussels. Zebra and quagga mussels have a basal thread, which our native mussels do not. So that's one way to tell. So if you ever see that, definitely let us know. Then we have New Zealand mud snail. It's the one next to the penny. These things are 5 millimeters to 6 millimeters in length. So they are very, very tiny, and they can pack into high densities within a square meter over 1 million. So the numbers are just crazy on those. Then we have African cloud frogs. This is a big one with the pet trade. They're very cryptic, stick to the water, and anything they could get in their mouth, they'll eat. So they're like bullfrogs, but a little bit more, stay in the water a little bit more. And finally, we have a European green crab. It's got the 5 spines on either side of the eyes, and that's one of the best ways to indicate or to identify a European green crab. So these are the 4 that I'm going to be talking about. Next slide. Our top priority, we want to find these zebra and quagga mussels. As already stated, there's lots of economical and ecological implications if they were to get into our systems. So next slide. In 2020, we do mussel monitoring throughout our state. And in 2020, we visited 442 sites, and that's 268 water bodies. So these are all our sites. Each site we have on a ranking system. So we'll visit a site 1, 2, or 3 times a year. That's all based on the risk that we'd encounter. It'd be how good is the boat ramp, how often is it visited, is there a boat ramp at some of these locations? Some of these might be just for kayaks. So we try and take all that into account to build our risk assessment. Next slide. Next slide. There we go. So what are our methods? We have artificial substrates. We do vertical and horizontal plankton tows. We do a visual shoreline survey. We look at water quality. Calcium, dissolved calcium is a big one for us. If they don't have the calcium to build the shells, they won't survive. So the more calcium a water body has, the higher likely that they'll be to survive and take off, you know, those exponential reproductive numbers that they have. And then we also implemented environmental DNA a couple years ago and Ponar grab. With the environmental DNA, it's kind of nice because we could actually take a water sample and look for multiple species. So we have included zebra quagga mussels, New Zealand mud snail, and pike into our eDNA sampling, which is kind of cool. Next slide. So these are our artificial substrate. It's PVC plates that are tiered. These are basically artificial homes that we'll put on the end of docks, buoys, next to boat launches. And the idea is the boat will be launched, they'll fall off into the water and hopefully settle out onto these artificial substrates. So in 2020, you can see that our numbers have increased. So we're trying to get more monitoring visits, trying to get to as many water bodies as possible, along with the number of sites. And thankfully, we have some partners. We have Chelan, Douglas, Grant County PUDs, and the Spokane Tribe. They help us monitor and do some of these monitoring methods to look for these guys. Next slide. So the vertical and horizontal plankton tows, we use a plankton tow net. It kind of looks like a windsock with a catch basin on the back. So we'll drag that through the water and get our horizontal, and then we'll drop it down to the bottom and then pull it back up and try and catch those veligers, the juveniles in the water column. And then we'll send that off to be analyzed to see if there's any of those veligers in the water. And again, 2020, you can see our results are up on that. Next slide. A quick, easy one that we can do is a visual shoreline. So we'll just walk the shorelines for about 10 minutes either side of a boat ramp. We look at buoys, we look at cement pilings or wood pilings, cement blocks, anything and everything that's on the shoreline. We look for zebra and quagga mussels. Lots of times we'll actually see other invasive species. We'll see Asian clams. A lot of the times we'll see crayfish, invasive crayfish, like the northern crayfish. So this is a nice, quick, easy thing that we do to survey for these as well. Go ahead and next slide. So water quality, we look at visibility using a Secchi disc. So we drop that in the water, kind of gives us an indication of how many particulates are in the water, how far down we can see. We also look at salinity, pH, and temperature. Temperature is a big one for their reproductive. Salinity is a big one. Some of these saltwater estuaries and stuff, you know, that brackish water could potentially hold some of these zebra and quagga mussels. So the higher the salinity, the less likely they'll survive. So if we could determine that on some of these water bodies, it's all good information to have. Next slide. And dissolved calcium, touched on this already, but the more calcium that's in the water, the easier it is for them to make their shells. So if a water body doesn't have that, you know, it won't be able to support them. So this is a great way to determine if their survivability would increase. And again, you can see our results are up for 2020. Next slide. eDNA, we pump water through a filter and send that filter off to a lab in-house. at WDFW, and we look for northern pike, zebra quagga mussels, and New Zealand mud snail. So it's a nice, quick, easy way to look for multiple species that could be potentially there. If we do get a hit, doesn't mean that they're there for sure. That just means that DNA is found. So it's nice because Grant County and Douglas County, they also look for northern pike and New Zealand mud snail and zebra quagga mussels. So our numbers are actually down on this because the price to operate this has gone up, and it is a little bit more of a time sink to do this. So pumping that water takes a little bit of time. So we actually decreased the number of samples that we take for eDNA. So next slide. So our last monitoring method is Petite Ponar grab. So you could think of this as the stuffed animal claw machines that you spend 10 bucks on to get a 50-cent stuffed animal. You drop this down to the bottom of to the substrate, engage it, and then pull it up. And off to the right, you can see that we pulled up a bunch of Asian clams or corbicula in that sample. So, you know, you can imagine if this was zebra or quagga mussels, if they are attached to rocks or down in the substrate, we'd be able to scoop up a bunch of them and detect them that way. And this is another one that our results have gone up in 2020 as well. Next slide. Thankfully, we have no zebra or quagga mussels that were detected since starting this program. Like I said, we do see a lot of different things, like northern crayfish down in the bottom right. We have a native clam on the bottom left, along with a invasive Quagga species, an invasive snail. And then we got some sponges, and I'm still not sure on what that red material is on that artificial substrate. So you can see how the artificial substrates work in this photo. Next, please. So Washington Department of Fish and Wildlife is the state lead and charged with planning, coordinating, and leading the implementation of management actions, including prevention, early detection, rapid response, control, eradication, and research. So how do we try and manage all this and prevent invasive species coming into Washington? Next slide. Number one is prevention. The more prevention that we could do, the lower the costs and the better it is for our economy and ecology of Washington. In this graph, you can see as the area increases of infestation increases, the control costs increase, and this is all on the bottom timeline there. So, you know, the sooner we can get to these things and get them out, the better it is. So with prevention, we do outreach and education. We also do decontaminations. The next step would be eradication. So we want to be able to treat some of these areas that they infest. One way is chemical, another way is trapping. And then containment. Containment would be the next step. Sometimes eradication and containment can flip-flop a little bit, but that is all part of our rapid response. When we get a report, you know, prevention has failed, we get a report about something, we go out, check it out, and then we try and figure out whether it's eradication or containment is the best option or both. And then if those fail, then it would be long-term management is what we would go into next. Next slide. So talking about prevention, outreach and education is one of the main things that we can do. The more people know, you know, to look out for these things, the sooner that we catch them, you know, that increases our chances and decreases our costs. So we do presentations and trainings like today. We go to sportsman shows, fairs, we create brochures and ID cards. We go to the boat ramps and put up signs. You can report these, any sightings that you may see. If you think that you see an aquatic invasive species, please let us know. RCO actually has a great reporting app. You know, if Justin could actually send that out to everyone, that'd be awesome. We also have an AIS hotline. So if you need a decontamination of your boat, you can give us a call and schedule an appointment. I'll talk about that in a little bit. And then social media, we've had things on Facebook, posts on Facebook and stuff like that about some of our events and what we're trying to put out there. Next slide. The next step in prevention is decontamination. The level 1 is one that everybody can do and should be doing when going to new places and exploring the outdoors. You want to clean off all macro stuff, you know, dirt, debris, any kind of invasive species that might be on your boots, whether it's plant material. So the idea is you just want to remove all organisms and debris. The next step is to drain. So any water body that you go to and you visit, you may get top your boots or something. You want to make sure that you get that water body's water out of your boot, and you want to make sure that you rinse it with potable water. You know, you want to use fresh water at the site to rinse any of that stuff off. And then the best thing you can do after that is dry it. These are a lot of times these are aquatic animals. Well, these are aquatic animals, so drying them out, desiccating them, you know, prevents their spread because they'll die off then. And this is a great way for terrestrial habitats too. If you think about going out and you're hiking around, you know, you pick up some cheat grass in your boot, you know, you don't want to take that to a new location. So you want to make sure you clean off your boots, rinse them off on site, and then dry them as well. Next slide, please. So here's some basic stuff that you could use for decontamination level 1. As a boot brush or a boot pick, you know, that macro material off. Then having some kind of potable water source. We have a bag with a hose on it that works really well. And then we also have a tote and garbage bags that we use. That way when we go from location to location, we could actually bag it up. Well, not location to location, but when we go from our location back to our home office, then we could bag it up, and then we know that we're not contaminating or potentially contaminating things along the way. So next slide.
And Richard, you've got about 5 minutes left.
Okay, I'll try and speed this up a little bit more. So then you know, gear must be decontaminated every day when leaving the area or water body. You want to do a level 1 before doing a level 2 decon. And then, like I said, having a bag or or tote with you is a great option before you do the level 2 to get it there and you don't spread the invasive species. Next. Quick plug, we're getting some of these. We have a pilot project going. These are decontamination stations that will be going out to boat ramps. And it's basically a big level 1 option for your boat. It's got a vacuum, brushes. Next slide. So level 2 used by professionals, and the objective is to eradicate and eliminate any of the remaining invasive species that could be on your gear. Next slide. We use hot water. It's 140 degrees for 15 seconds, excuse me. If you have anything like any boat or felt-soled boots, you want to make sure that you soak that for 5 minutes at that temperature. And this is environmentally friendly because you don't have the chemicals. Next slide. This is what we use. We actually have a hot water pressure unit that we could take to marinas and decontaminate some of these boats. So it's a nice option that we have available. Next slide. We also have decontamination stations. One is at WDFW Ephrata Regional Office, and the other is at the boat check station in Spokane. So that makes it a nice one-stop shop for deconning your boats when you're coming into Washington. Next slide. Another option for level 2 is chemical. We use Virkon Aquatic at a 2% solution for 20 minutes. And it's a great option to soak your equipment and kill off any aquatic invasive species that might be there. And then rinsing it off is always a must. So, next. So talk about this really quick is going into what we could do is containment and eradication. And thinking back to that chart. In 2015, we got a report of African cloud frogs at the 3 Lacey ponds. They're stormwater ponds, so they're man-made. And we worked with the city of Lacey to actually put up this silt fencing to contain them. Next slide. So the next thing that we thought of was, well, how are we going to treat these things? So we got Jackson Groves from Smith Root to conduct a salt pesticide treatment experiment. And he found that 12 parts per thousand is an effective treatment option to try and eradicate these guys. And we got approval for our pesticide permit. Next slide. And then we treated the smallest of the ponds, the 3 ponds. We drained the water levels, put a silt fence or another silt fence in that. That way we could try and get any additional escapements trying to leave the water, the lower water body. And then August 2017, we actually applied the salt treatment. We did 2 treatments over 3 weeks. And the results, we got 508 African cloud frogs, 2 bullfrogs, and 260 native species, and 9,670 goldfish. And that was a lot of goldfish. And some of these goldfish were actually about 8 inches long, or yeah, about 8 inches long for some of them. So it's kind of surprising how these goldfish got in there. And African cloud frogs, more than likely it was just a release of a pet. So next slide. So post-treatment, things were looking really good. In February, we found one specimen, excuse me. It was the largest gravid female that we've ever seen. And then in July 2018, we captured 28 juveniles. So it took quite a while to actually get to capture more. So we thought, okay, there might be a little bit more of recruitment for the species. And then maybe another treatment would work. Well, in October of 2018, the city of Lacey found them in their stormwater catch basins that lead to the stormwater ponds. And so that is just another source of recruitment that goes into these ponds. So even if we were to treat it again, we'd have to figure out what to do with those pipe systems because they could recruit into those ponds still. So now our next movement is thinking long-term and a more collaborative way of doing things. So long-term management, that's what we're thinking about next. When prevention is not enough, we have species that show more widespread distribution with an increased abundance that we aim towards controlling through long-term management. Although total eradication may be impossible to attain, some local site-specific eradication may be achieved with intense effort, but the department cannot do this on their own. Collaboration and coordination between co-managers, local governments, and stakeholders is crucial. One example of aquatic invasive species is the European green crab that's in this boat now. Green crab were introduced in Washington through larval dispersal from the outer coast, with the earliest recorded crab being discovered in 1998 in Willapa Bay. Since then, green crab have been increasing in abundance throughout the outer coast and spreading into the Salish Sea. Green crab don't follow human-derived boundaries, so fencing these guys off doesn't quite work that way, like we were able to do with the African cloud frogs. So we must work collaboratively and coordinate management efforts across all jurisdictions. The more coordination we have will enable us to enable us to leverage more resources, avoid competing for the same resources, or duplicate efforts. Currently, green crab does not have a cohesive statewide management plan, but that's something that we're working towards. We do have a collaborative management plan called the Salish Sea Transboundary Action Plan. Justin linked that earlier, that we do work with, and that was, And having issues with my computer. There we go. And we've been working with what's been the Washington Department of Fish and Wildlife, Puget Sound Partnership, Washington Sea Grant, and the Department of Fisheries and Ocean in Canada. Next slide. So green crab, as well as all or most other invasive species, fall on an adaptive management-based approach. This requires open two-way communication between co-managers, partners, and stakeholders so that we do not lose time in trying to figure out what is happening. We also want to disseminate effective strategies as quickly as possible. To do this, we must collaborate and coordinate management strategies to allow for all of us to achieve or share goals in protecting our environment, economic and human resources. So sorry that got a little rough. I was trying to rush a little bit, but with that, thank you. I appreciate your time. And back to you, Alexis.
Yeah, thanks, Richard. We'll go ahead and make sure to share everybody's contact information. And again, watch out for that recording that we're going to have posted in about a week or so. But before we get to questions, because I want to be mindful of everybody's time, for those of you that are here and hoping to get pesticide recertification credits, this is your code word. Your code word is puddles. So if you could please type the word puddles into the chat, that is how we're going to confirm your attendance for the day. And if you have another meeting you need to get to, as soon as you've done that, feel free to log off. But we are going to move into questions. So we're going to go a little bit over time today for those of you that want to stick around to get some of those questions answered. But again, the code word is puddles. So for those of you that are on the phone, you can email me later too, if you want to. But for everybody that is currently on the webinar hoping to get pesticide research credits, if you could type the giant red word into the chat, that is how we're going to confirm your attendance. And so I'm going to leave this slide up just for a minute or so, but I'm sure I've seen the little chat thing going off. So I know we got a bunch of questions. Justin, would you mind going to maybe the first one, and I'll leave this slide up as we answer them.
I'm sorry, are you asking me to ask the question?
Yeah, can you ask the panelists the first question? I can't see it quite yet.
I'd be happy to.
Thank you.
So the first question that we have is directed toward Richard, but feel free, Captain Anderson or Amanda, to jump in. For your vertical and horizontal toes, do you combine into a composite?
So what we do is, yes, we take our vertical toe, we place that into our sample bottle, and then we'll take our horizontal toe and place that into the same bottle, and then preserve that, and then ship that off to get analyzed.
Great, great.
Yeah.
Another follow-up question related to monitoring. In order to reduce cost, can't you do unfiltered samples, such as preserve according to lab procedures for analysis?
Is that referring to eDNA?
I believe so.
Yeah, there are methods to where you could actually scoop up water and then freeze that to certain temperatures and get it sent off to analysis. So that is an option, but because we're traveling all throughout the state through different temperatures and stuff like that, we're not always going back to a home base where we have a freezer always available. We're staying in hotels and stuff like that. So the best thing that we do, I feel, is using the filters, because we use desiccating beads to take off the water and preserve the samples that way.
Great. So I've got a question. I think it is directed to the good captain. You showed a map of the watercraft inspection stations, but do you partner with other organizations that also do watercraft inspections within Washington?
Yes. Currently, we have a memorandum of understanding with the National Park Service for Lake Roosevelt rec area, and they do inspections in conjunction with us. Sometimes we send Sergeant Taylor up there to work with them, or they run them on their own. Also, in the Spokane area, we have the Diamond Lake Homeowners Association that has partnered with us, and Sergeant Taylor has trained a bunch of the homeowners, and they run inspection stations there and give us the information of how many boats they inspect. We also have a MOU currently with the Clark County Sheriff's Department to conduct some inspection stations down there. I haven't heard from them based upon COVID, what they did last year. Then we also are working with state parks. There was a question in the chat about boater education. So currently we've been working with state parks. Aquatic invasive species is a section in the recreational boater card requirement course. And then they also are utilizing, state parks runs the BMLE, which is the basic marine law enforcement. So all the county law enforcement and municipal law enforcement officers that are around the state doing safe boating inspections have also been trained in aquatic invasive species inspections. So they're working from that end. And then finally, I know that Whatcom County also has a inspection program up on Lake Whatcom, and we've been working with them as well. So we are expanding the groups that we work with, and the more eyes and ears and hands and everything that we can get checking boats in Washington, the better. And then Justin, you want me to snag this last one from Joseph Shea about the point source infestations and water shoreline owners?
You're the boss. Take it away.
Okay. So currently, that's a big question. I've read it, and the current answer is no. There's actually no enforcement protocols for shoreline or landowners in relationship to zebra quagga mussels or the other types of aquatic invasive species, because they actually can move back and forth, and we have nothing in place. There is the weed board, noxious weed control board, because there are the aquatic vegetations that are also considered AIS, and they deal with that. So at this time, long verbose answer, no protocols. Cross that bridge when we get to it. That makes sense.
Thanks for that. And we also had a question in the chat about inspection of commercial ships. So can you speak more to that, Amanda?
Yeah, so for large commercial vessels and like the merchant fleet that's coming to Washington, so I'm actually the data manager for the ballast water program, which is a little small part of the AIS unit. So we deal entirely with commercial vessels. And you can imagine there's a lot of challenges associated with getting on board one of those vessels and being able to inspect directly, but we do have an inspection team. It's 2 inspectors that cover the whole state, and they do their best to get a representative sample of the vessels that are coming in. For every vessel that arrives to Puget Sound, or the Puget Sound, the Columbia River, and the open coast, we review paperwork for them, and I review like their positions when they were doing different management methods for their ballast water and for biofouling, and see if they did that correctly. And then our 2 inspectors go out and try to get a representative sample of vessels. And what they do is go more in depth. We've got a Chief Engineer Gary Gertson, and Captain Keith Strike are our 2 inspectors for that. So they are subject experts. C.E. Gertson is a former merchant vessel chief engineer, and Captain Strike is a former U.S. Coast Guard captain. And so they have a lot of experience boarding vessels, looking at the operation of vessels, looking at the management logs that vessels are using, and they are very familiar with what the engine rooms and all of that look like. So they go and look primarily at, is the vessel following best practices.
Great, thank you. And this will be one we'll pass around to each member of the panel. But do you have examples of success of your program's accomplishments? And I'll note this is challenging in invasive species, because if we have adequate prevention and policies, you don't really see the invasive species. But I think you have many examples of different successes, and if you could each take a minute or maybe up to two to convey some of that, please do.
I guess I'll dive into this one. I put the success, at least of the Watercraft inspection program, if you remember back on the presentation I did, is the growth in the number of vessels that we are able to inspect now. Honestly, keeping aquatic invasive species out is kind of like being a goalie. You're trying to keep the points from going, or the ball from going in the net and scoring the points, and it's sheer numbers game. And if you think about it, we inspected over 32,000 boats, and we had 25 boats that we had to deal with that had zebra and quagga mussels on it. So that's a success. There were 25 shots out of 32,000 on the goal that we were able to keep out. And so that's a success. If it takes one lightning strike and we're done. And so the more that we can grow the program, the more we can do those inspections, the more likely we will intercept those boats and keep them from being out of the water. And then the other success that keys into that is, like with Richard's program, they haven't found them. So, that's the biggest success I could see, is if we monitor and we don't find them, we've been successful. So,
yeah, I definitely agree with that. And to add on that, we actually did a rapid response exercise. And with that rapid response exercise, we used our mussel monitoring techniques. We used Canadian and puddles dogs to try and sniff out zebra and quagga mussels that were planted. We put out a boom that would be a containment option, and treated the water with dye. So that was like our chemical treatment. And we actually had divers go out as well. So we actually implemented all this over a week, and I thought we did a great job doing that. So, that's another thing that we have in our tool belt right now.
So there's a couple of things. A success story from, say, my perspective in the ballast water program is when I think I showed a picture of Captain Strike taking a ballast water sample. He was dipping one of those nets down into the ballast tanks. And that was during a study that we did with Jeffrey Cordell's lab at the University of Washington. And Captain Strike was on an articulated tug and barge, and those are a very specific and very risky kind of vessel that operates inside of Washington State and Oregon. And the reason they're risky is because they go on very short little voyages. And so anything that they're picking up down in California, it wouldn't have to live very long to get here. And they do, these are crews that are very interested in doing their job safely, doing their job in accordance with the law. So what actually happened was Captain Strike set up a little competition, because he would get back the results from Jeffrey Cordell's lab that would tell how many organisms were still in their ballast water after they had done a certain management technique. And so the vessels were starting to share information with each other and say, hey, we did better than you. We only had like a couple of little organisms in our ballast water and no identified invasive species and stuff. Like, oh, well, hold on, we're going to do better next week, And so it was this friendly competition that started among these vessels that are very, very specific to Washington's waters, and who we really want to work closely with because they live right here. And they also, just like us, are hikers and people who like to go fishing and people who just love Puget Sound and the Columbia River in general. And so they understood both the reason that they were doing these things, and they started to really see the results of this additional management of their vessels that they were being asked to do, because at first it's kind of like, oh gosh, it's another thing, right? But they really started to have fun with it. And that was a real big success story. If you want a specific success from an eradication effort, this is a little bit of an older example, but it is from California, and it involves the invasive weed the invasive alga Caulerpa taxifolia. And it's more of a general example, but it is an example of what absolutely has to happen for an eradication to be successful. Divers found this alga at the bottom of a tiny little enclosed body of water. They immediately knew the scientists to alert. They alerted them. They got a positive ID. I knew the scientists that were working on this. It was a little bit before my time at the lab that I was at, but they were immediately told. Everyone immediately went in. There was state funds were already available, said if you find this weed, this money gets mobilized. The money was immediately mobilized. They put tarps down over these alga and put bleach under those tarps and just, scorched earth the little patches that were found. It was completely eradicated, and Caulerpa has not come back to that bay. This is an example from the early 2000s. It is, outside of Washington, but it is an example of why we do so much prevention and why, those eradication efforts are so difficult. I mean, imagine if that had been, that Caulerpa invasion had been allowed to go for a few more weeks, months, or maybe a year. There would have been no way for that to be as successful as it was. So, all that money and all that, all that infrastructure had to already be there. Everybody had to know what they were looking for and know exactly what they were going to do when they found a patch. So you can find that online, and I'll type in the chat the name of the alga that is. That was down in California in the early 2000s again. So it's just an example of like why we do prevention, because this was already still, even that tiny little patch cost millions of dollars to get it out. So.
That's great. That's a great segue. I feel like to the end as well, because that's really what drove us to work with Governor Jay Inslee to proclaim Invasive Species Awareness Week, because as you can see, we've got all these great individuals working on this, but it's a really big problem with many different pathways. And we really do need everyone to be aware of invasive species. And if you see something, say something, because it may be the very first patch of Caulerpa. It may be the first Quagga zebra mussel that happens to sneak through our front lines. It could be any number of things. And if you notify us, then we have that opportunity to respond and take that shot, as Captain Anderson says, in terms of stopping it.
Hey, Justin, one quick one. There was a question that came in about if there was collaborative management efforts with adjoining states and Canadian provinces. And simple answer, yes, on that. There is a great deal of collaboration with our other state agencies. We're part of the Pacific States Marine Fisheries Commission 100th Meridian Group, and there are quarterly meetings, and the state invasive species coordinators regularly meet, currently now during video or phone. And particularly the Pacific Northwest has very similar programs when it comes to watercraft inspections and monitoring programs. So yes, this is a concerted effort, because it can't just be one person or one agency doing it. It's a regional effort. And you can find more on, what's that website? Go to the Invasive Species Council. I think we got a link to the Western AIS as well. So sorry about that.
It in fact is Western AIS is in Aquatic Invasive Species, but the acronym westernais.org organization.
And from the Aquatic Invasive Species Unit website, you can also find interstate and international information on our ballast water program. So those merchant vessels, if you're interested in that level of stuff, we're part of the Pacific Ballast Water Working Group, and we're also part of a federal group with the U.S. Coast Guard and the U.S. Environmental Protection Agency.
Great. Well, we still have 54 participants, and the questions are still rolling in. So as long as people have questions and you all as panelists can stick around, I say we just keep going. This is such a rare experience to convene this great group. To Richard, we have a question from Janet, and can you address the use of other decontaminants, for example, that can be used for boots? They have used a QAT chemical solution coming off of an Oregon spotted frog site in Whatcom County, and they've also used bleach solutions between amphibian survey sites. So I guess, can you speak more to that level 2 decontamination sounds like? Excuse me, got my dog out.
For level 2, lots of times, like I said, we use Burcon and hot water as our 2 main ways of doing it. And me personally, that's what I've used since starting in this program. So I don't know much about quat or other chemicals out there, to be honest with you. I'm sure there's other options that work. You just have to follow the labels. It's the best I can give you on that one. I just don't have the exposure to the other chemicals right now to be able to address that very well.
Thanks, Richard. Sounds good. Justin, do we have any more questions? I think we got most of them.
I think that was all the questions. I did have one question for Richard. You showed the decontamination systems, the one that's in Ephrata and the one that's in Spokane. If folks were interested in scheduling a decontamination, how would they go about doing that?
Yeah, they could call the AIS hotline, and I believe it's option number 3 will take you to that. So you could schedule it that way. So I don't have the number off the top of my head, but I could snag that and send it into the chat here if you give me a moment.
Oh, Eric, you're muted.
If I remember off the top of my head, I think it's 1-888-WDFW-AIS. I think I'm pretty sure that's the number.
That
right.
sounds
I was
about
looking
right now that
it.
I think about it. Yeah, I was trying to think of the actual numbers, but that's easier.
Actually, I got to make a comment about rapid response, and again, particularly in the case of zebra and quagga mussels, you know, there's the old saying, an ounce of prevention is worth a pound of cure. Honestly, an ounce of prevention is worth, you know, billions of dollars a cure with it when it comes to zebra and quagga mussels. And I hate to say it, we're preparing for rapid response, but God, I hope we don't ever have to use it. That's the biggest thing. You prepare and hope that you don't have to use it. Now, I'll put it this way: if anybody wants to, you know, again, for the planets to come completely into alignment for a rapid response eradication to work, I want everybody to take a can of whipped cream, take all the whipped cream out of the can, and then try to put it back in. Okay, that'll give you an idea on how successful it would be to try to put these things back in the can, to put Genie back in the bottle. Because, you know, yeah, you can kill something in the lab, you can in a controlled site, but if you're trying to do chemical applications and containment in a river system, just think of all the variables that come in: water flow, temperature, traffic, number of boat ramps. Again, I mean, that shot to do it and get it out of the water is, again, when Powerball. If they get in, we're going to live with them. So again, prevention is the key on these things. Not to be a fatalist, but that's the main thing. I'm a realist, is what I say. Keep them out, because once they get in, you're probably going to live with them for the rest of our lives and then on.
I hadn't heard the whipped cream analogy, but that is the best analogy I've heard. It's like, you might be able to do it given time enough and resources enough, but the likelihood is not, the odds are not in your favor. So that's the best analogy I've heard, Eric.
Might be able to, but I doubt it. So
well, that said, I just put a link in the chat to a recent eradication that was successful in Texas. But the key was that it was found very early. And so that's just a call back to action to everyone on the meeting. If you help us find it, or if Richard's monitoring team finds it quickly, we do have a shot, and we shouldn't give up immediately, Captain.
I'm not saying give up. I'm saying have fun trying, but I want everybody to go try to put whipped cream back in the can today. I think they would have a ball.
That's your secret motive behind all this. You just want to see if people are going to do it. Well, perfect.
Tag me on Instagram.
Right. Tag the DFW account. I don't see any more questions rolling in. So if anybody had any last comments, we'll give it just a second, but I think we're ready to wrap up. And I want to just thank all of you so much for coming and lending your expertise and talking a little bit more about your program. I think this is really important and valuable information that maybe the broader public isn't aware of all the great things you're doing. So thank you, each and every one of you.
Thank you for inviting us. So thank you.
Yeah.
Seeing the level of interest in these talks is really heartening. I think people are, I'm just so glad to see that people are aware this is an issue they want to be informed about.
For sure. Perfect. Well, with that, happy Wednesday, all. Happy day 3 of Invasive Species Awareness Week. We did it. Yeah.
Thank you all.
Thanks, everybody.
Bye.
Thank you. Have a good one.